So hello everyone, my name is Isela Hernandez. Our project is drug reactivity via DNA sensors, and our mentor is Jacob Summerson.
And like she said, my name is Ken Chao.
My name is Ina Santiago.
My name is Electrode.
And again, I'm Isela Hernandez. So cancer is a disease that leads to an uncontrollable growth and spread of abnormal cells. A treatment process known today is chemotherapy. A drug used in chemotherapy is doxorubicin, which we have a picture of here. Doxorubicin is injected into the patient's bloodstream, and it helps stop the replication of DNA. Once the drug is injected into the patient, it's hard to tell how much how much would be beneficial for the patient because it depends on height, weight, and health of the patient. Here we have a graph of when the drug is initially injected to the patient, and the concentration of the drug spikes, and then the body gradually breaks down the drug. If the body doesn't break down enough of the drug, it is fatal for the patient. And if the body breaks down too much, then the cancer cells aren't killed. So a method that doctors use today to evaluate these recordings are they extract multiple blood samples at varying times from the patients, and they look at those, and which isn't always, it's not an accurate method, and it's very timely. Our mentor is researching a way for a DNA sensor to be injected into the patient to record and send accurate readings in real time, and this will ultimately benefit cancer patients.
So our contribution to our mentor's goals was to optimize the effectiveness of our sensor, and the two mediums that we were using were buffer and blood. We used buffer because there will only be a direct interaction between the drug that we add and our sensor, and we are using blood because blood is the medium that we wish to work with eventually. In our buffer, we are looking for two things: signal gain and sensitivity. And signal gain is the percent increase seen when a drug is added. And we're also looking for sensitivity, and sensitivity is how well our sensors can detect concentrations of our drug. In blood, we were looking for two different things. We were looking for the robustness, and we were looking for the signal survival. So robustness is saying, in blood, our sensors act differently than in buffer in the fact that there's a decay in our signal change. So we were looking for how well our test sensors decay when we put it in the blood. And we were also looking for signal survival. So obviously, since the signal is decaying over time, we had to test whether or not a signal change can occur when we edit our drug. And here we have this graph is just a representation of our test in buffer, which is signal gain and concentration. And the difference between one point of our readings and another point of our readings is essentially our signal gain.
So in our experiment, there's two important elements to consider. First, the synthetic DNA. So what exactly is a synthetic DNA? A synthetic DNA is an artificial DNA. So our mentor used a specific type of DNA. This specific type of DNA bonds to the drug that we're looking for. The second important element is the electrode. This electrode has a golden surface. The golden surface is important because it bonds to the DNA as well. So I would like to introduce to you how we began our procedure. First, we mechanically cleaned our procedures. It is important to clean your procedures because it prevents from interference to occur. Secondly, we molecularly cleaned. By this, we molecularly clean because there's particles that we can't clean manually, so we have to oxidize and reduce it to clean it. Third, we prepared our DNA. When we prepared our DNA, we got our DNA and mixed it with T cell. This T cell allows the DNA to break in half. At the end of DNA, there's sulfur. This sulfur bonded to the gold, which was on the electrode, allowing the electrode and the DNA to be together. So that's the DNA insertion. Okay, so when we made our electrodes, 12 out of 6 out of those 12 had higher concentrations, and the other 6 had a lower concentration. By that, I mean 6 had double the amount of DNA on the surface of the electrode, while the other one had half the amount of the double.
So after we have coated each electrode with different concentrations of DNA, we are now going to take each one and test it for how well it conducts an electrical current. So we use here a potentiostat, and what the potentiostat does, it accurately measures and controls the electrical current. So we are taking 6 sensors that are being made out of high concentration DNA and the other 6 that are being made out of low concentration DNA. And what we want to do is we want to test each one individually to see how well they can conduct the current. And after we have tested them, we're going to select the top 3 to see. We're going to select the top 3 from each category to see how well they can conduct, how well they can detect the blood. So here we have the interaction process. As you can see right here, we have the electrode right here, and we have the DNA that has been settled on the surface of the electrode. On the top end is methylene blue, which is electrically active, and here we have our target, which is doxorubicin. So what we want is an interaction to happen between the DNA and the doxorubicin. And in order for this interaction to happen, we need to send a voltage and increase the voltage to a level where this interaction is going to happen. And the interaction is going to cause for the DNA to recognize the doxorubicin. So once it has recognized the doxorubicin, the DNA is then going to fold up, aligned with the methylene blue here, to come near the surface of the electrode, which will increase the number of electrons being transferred. The number of electrons being transferred will send a signal through an increase of current, and this signal tells us the amount of drug that has been detected.
First, when we tested our electrodes and buffer, we found that the sensitivity, which is the lowest amount of concentration detected on our sensor, was recognized in both high concentration and low concentration. And our and signal gain, which is the highest percentage found when drug is added, was favorable in the low concentration. When we tested it in blood, we also found that the low concentration sensor was more favorable for signal survival. So overall, our low concentration sensor worked better in buffer and in blood.
So, as Estella previously explained, the M buffer, as Estella previously explained, M buffer, the low concentration had a better signal gain than the higher concentration. You can see this through this graph. As you could tell, the lower concentration was always higher than the one with higher concentration, therefore indicating that it had better signal, that the lower concentration had a better signal gain. Also, by the curve, we could interpret sensitivity. As you can tell, both of the curves started at the same spot. Therefore, the sensitivity were the same. But if they were not the same, one of the curves would be more to the right because it would detect the concentration at a later rate.
So this is the data that we collected when we put our sensors in blood. As I said earlier, blood reacts differently with our sensor and the fact that there's a signal decay instead of a signal increase. We believe that this is because our current theory is that there is something interfering in the blood, interfering with our sensors, and it's causing this decay. So this region right here is that decay. This region was before we added any of our drug, and we just let the signal decay over time. And as you can see, the low DNA concentration sensor decayed at a slower rate than the high DNA concentration sensor. And this, we can conclude from this that it's more robust. This allows to have a longer longevity. And when we added our drug, we noticed that there was a high increase in the signal change, which is about, from the graph, about 40% for each sensor. Now, we can't have any definite trend. We can't conclude any definite trend from this, from when we added the drug, but we can conclude that there is a signal change that is only due to when we added our drug, and from that signal change we can find the current, and then we can find the concentration from the graph that we used, that we created when we're testing our drug in just buffer.
So what have we covered and what does the future direction of the DNA sensor look like? Well, first we started by testing the DNA sensor in buffer, which we addressed signal gain and sensitivity. Then we tested the DNA sensor in blood, where we're going to address signal loss. We weren't able to do much for that, but our mentor is going to work on providing a solution for this. He is then going to test the DNA sensor on a live animal, where he's going to also address any issues that he finds. After this process has been completed, the DNA will now be ready for clinical trials and will be on its way to make cancer patients' treatment easier. We would like to thank Jack and Cooke Bridges program for providing this internship to community college students.
We'd also like to thank the California Nanosystems Institute for accommodating us this whole week and for providing us with a lot of valuable information that we'll use in the future.
We would like to thank our wonderful RAs, or resident advisors, who assisted us and guided us throughout the whole week.
And we'd also like to thank Dr. Kevin Plaxco for allowing us to work in this lab, and especially our mentor, Jacob Summerson, for his patience and guidance throughout this whole week.